Hi everyone, my name is Alexandre Carbonneau, and I'm here to present you the project I am currently working on, which is a part of the Pan-Canadian project called FISHES for fostering indigenous small-scale fisheries for health, economy, and food security. And more specifically, the title of my project is Population Genomics of Ungava Atlantic Salmon and Implications for the Management of Subsistence Fisheries. So let's start by mentioning that Ungava's Atlantic salmon populations are of great biological importance, particularly because they are the most northerly populations in eastern North America, and also because of their diversity of migration patterns. But most importantly, these populations contribute to the economic, cultural, and food security of Ungava Inuit communities, since many salmons are fished within the Coxswack River estuary, as well as within each major salmon rivers in Ungava, which correspond to the 7 watersheds represented by the 7 colors on the map. Unfortunately, there are only a few studies that have addressed this system, which means that there is a lack of information to properly manage the salmon fishery in Ungava, which compromise the long-term sustainability of these populations, especially as there has been a significant decline in salmon stocks since these studies were conducted in the early 80s. Thus, the first goal of this project is to partially overcome this data gap by documenting the genetic structure of the 7 major salmon rivers of the Ungava. In other words, I'm aiming to document the genetic differences between Ungava's 7 main salmon rivers. To do so, a part of the adipose fin was taken by the Wildlife Ministry and by sport fishermen from about 40 salmons in each studied river in order to eventually extract their DNA. And in its simplest form, DNA consists of a long sequence of 4 different molecules, represented by letters here. Since this sequence varies between 2 individuals, it is possible to differentiate them genetically, and it is therefore, by extension, possible to differentiate 2 groups of several individuals from these same dissimilarities. Once the genetic structure of the population has been clearly identified, the second objective of the project is to establish from which river the salmons caught in the estuarine fisheries of the Coxswack River originate. To achieve this, we are counting on the involvement of Inuit fishermen to collect samples from the salmon they net fish within the river estuary. These fishermen collect the adipose fin in addition to some other data, and then we proceed in much the same way as we saw previously to obtain the genetic data and eventually determine the river of birth of each salmon caught within the estuary. In conclusion, the global objective of the project is to improve knowledge of Ungava salmon in order to improve its long-term management and to maximize the sustainability of the fishery for future generations. Thanks for watching, and if you have any questions, please do not hesitate to contact me.